Okay, so today let's slow things down a bit because today on Shey's Microbiology, we'll be talking about gram-positive bacteria. And I want you to imagine you are in the lab with me, looking into the microscope, thinking through what you are seeing, not rushing. We will talk about gram-positive cocci, then move fully into gram-positive rods, but in a way that actually makes sense. So, let's say you've done your gram stain. You focus the microscope and you see purple bacteria. Already, that purple is telling you something important. It means thick peptidoglycan, strong cell wall, gram-positive. But then, the next thing your brain should do is pause, because shape matters. Are they round or are they elongated like little sticks? That question alone splits your entire thinking. So first, cocci, round bacteria. Now, here's the thing many students rush through. Arrangement, don't rush it, because when cocci divide, how they stay together matters. If you see them piled up, kind of irregular like a bunch of grapes, your brain should softly go, Staphylococcus. But if they are lined up like beads on a string or in pairs, then now you're thinking Streptococcus or Enterococcus. You haven't done any tests yet, but you are already narrowing things down. So let's stay with Staphylococcus for a moment. Clusters, purple, cocci. The very first thing we usually do in the lab is a catalase test. This test is so simple but so powerful. You add hydrogen peroxide and if it bubbles, that's catalase positive. Staph loves catalase, Strep doesn't. Already, one drop of reagent has separated two big bacterial groups. Now, within Staphylococcus, this is where people start memorizing names, but let's not do that yet. Let's think behavior. Staphylococcus aureus, for example, is aggressive. It produces enzymes, it produces toxins, it invades tissues. That's why it's coagulase positive. It literally clots plasma. So when coagulase is positive, the lab tech doesn't smile. They get alert. Because Staph aureus can cause abscesses, wound infections, pneumonia, even sepsis. Then you have coagulase negative staphylococci. These ones are quieter. Staph epidermidis lives on the skin, usually harmless until there's a catheter or implant. Staph saprophyticus shows up in UTIs, especially in young women. Same shape, same stain, different personalities. Now let's move over. Imagine your catalase test was negative, no bubbles. Now you are in Streptococcus territory. The first thing we usually look at here is blood agar, and this is where hemolysis becomes your guide. Clear zone, that's beta hemolysis. Greenish zone, alpha hemolysis. Nothing at all, gamma. Each one tells a story. Streptococcus pyogenes, for example, beta hemolytic, is behind sore throat, rheumatic fever, kidney complications. Then Streptococcus pneumoniae, alpha hemolytic, optocin sensitive, major cause of pneumonia and meningitis. And suddenly, these purple circles start connecting directly to symptoms. Now, let's pause here, because this is where many people mentally check out. Gram-positive rods, but this group is fascinating. Now, instead of round bacteria, you're seeing rod-shaped purple organisms. The first thing you ask yourself is, do these form spores? If the answer is yes, you're probably dealing with Bacillus or Clostridium. Bacillus species are usually aerobic. Think Bacillus anthrax, large rods, non-motile, very distinctive. Then Bacillus cereus, food poisoning, especially reheated rice. Now Clostridium. These ones are anaerobic. They don't like oxygen and they produce some of the most powerful toxins known in microbiology. Clostridium tetani, muscle spasms. Clostridium botulinum, flaccid paralysis. Clostridium perfringens, gas gangrene. Clostridium difficile, antibiotic-associated diarrhea. Not all gram-positive rods form spores. Corynebacterium diphtheria, club-shaped, arranged like letters. It doesn't invade deeply, it releases a toxin. That toxin shuts down protein synthesis, deadly if untreated. Then there's Listeria monocytogenes. Small rods can survive in cold environments. That's why it's linked to refrigerated foods, and in pregnant women, it's dangerous. And finally, organisms like lactobacillus, normal flora, acid producers. Not everything in microbiology is an enemy. Some bacteria protect us. So when you look at gram-positive bacteria, don't rush to names. Think shape, arrangement, oxygen preference, spore formation, biochemical behavior. Every answer leads to the next question. That's systemic bacteriology. And once you start thinking this way, microbiology stops being confusing. It starts being logical. That's gram-positive cocci and rods explained. This is Shey's microbiology, not just learning bacteria, but learning how to think like a microbiologist.